Welcome back everyone, my name's George, and today we're going to be extracting DNA from strawberries. What makes you you? What separates people from dogs, cats, dinosaurs, or even bananas? The answer lies in our DNA. DNA, or deoxyribonucleic acid, contains the genetic code for everything in our bodies, from what we look like to how our bodies work. It's basically a huge instruction manual for how our cells are supposed to behave to make everything in our bodies work correctly. These instructions are made of the letters A, C, G, and T, called nitrogenous bases, whose different patterns make a special code that is different for every living thing. DNA is so tiny that scientists need to use a high-powered microscope to see a single strand. But today, we are going to use everyday household materials to see DNA with our own eyes.
First, place the isopropyl alcohol in a freezer. Second, measure 1/3 of a cup of water into a small glass or container. Third, add 2 teaspoons of dish soap to the water. Fourth, add 1/4 of a teaspoon of salt into the mixture and stir until the salt dissolves. Here you have finished your extraction mixture. Fifth, place 1 strawberry in a Ziploc bag. We recommend you cut it up. Sixth, place the extraction mixture into the Ziploc bag containing the strawberry. Seventh, remove as much air as possible from the bag before sealing it tightly. Eighth, use your finger to mash and mush the strawberry in the bag so no big pieces remain. It should be a pretty liquidy mixture. Ninth, pour the strawberry mixture through a strainer into a glass container and use the measuring spoons to mush the strawberries so any excess juice is squeezed out. 10th, transfer the strawberry mixture to a small container that holds around 1/4 to a half cup of liquid. 11th, add 1 teaspoon of the chilled isopropyl alcohol to the mixture. 12th, you should see a separation of liquid in the container with a white layer on top of the liquid. This is the strawberry's DNA. Use tweezers or chopsticks or anything like a fork to pick it up and observe it.
Wow, how can you see the strawberry's clear DNA? What makes it look so much like a blob? What you're looking at now isn't the DNA from a single strawberry cell, but from the millions of different cells that make up the strawberry. But why did we decide to use a strawberry? To answer this question, we need to talk about chromosomes. Chromosomes are made of DNA strands wound like string around a protein. They hold your DNA in an X-like shape. Without chromosomes, your DNA would look like a messy pile of spaghetti. Each of us have 23 pairs of chromosomes in each of our cells. In a pair, 1 chromosome comes from your dad and the other from your mom. That's why you look so much like your parents. While us humans have 2 copies of each DNA chromosome in our cells, strawberries have 8. That's a lot of DNA. That's what makes strawberries perfect for this experiment. So, how did the experiment work? The secret lies in the extraction mixture. First, the dish soap dissolved the membranes holding the nucleus together. This released the DNA into the solution. Next, the salt broke apart the chromosomes holding the DNA together, which turned it into the messy blob that you see now. Finally, the isopropyl alcohol caused the DNA to precipitate or solidify because DNA cannot dissolve in alcohol, especially when the alcohol is freezing cold. So all of the DNA condensed into a single blob-like structure that seemed to float on top of the strawberry solution. If you want to extend your learning and do this experiment again, try using different fruits besides the strawberry. Bananas and kiwis work well. Also, you could try this experiment using your own DNA. Use a Q-tip to swab the inside of your mouth and follow a similar procedure to see strands of your own DNA. A link to video instructions on how to do so is attached below. One really important scientist who was largely credited with discovering the structure of DNA was Rosalind Franklin. She was an English chemist who used X-rays to allow her to see the molecular structure of DNA molecules. The photographs she took of her work later led to the discovery of the double helix ladder-like backbone that all DNA molecules are composed of. While she died before she could have received a Nobel Prize for her work, her partner continued her research and later received a Nobel Prize in Chemistry in 1982. That's all for today. Come back tomorrow to launch a soda bottle rocket using Alka-Seltzer tablets. See you there!